Thank you very much. Thank you very much for having me. Today I'm going to present some preliminary results from my master thesis study called Spatial Ecology of the White Seabream in a Small No Taxon. So to start, I would like to introduce some background information about my study fish species Diplodus sargus. It is a very highly targeted species by both commercial and recreational fisheries, which makes it vulnerable and in need for protection. As other reef-associated fish, it is characterized by a high site fidelity. Both male and female are sexually mature from January to May in this region. Reproduction is known to be triggered by a temperature between 15 and 17 degrees. It is also known that this species expands its home range area and foray more frequently during the reproductive season. And what is also important to say is that they reproduce in rocky outcrops in deeper areas than usual, according to the literature. Regarding the main aims of this study, first of all, we aim to study the movement pattern inside the no-taxon during passive acoustic telemetry. Then we decided to calculate the home ranges area and the core areas in order to assess the potential of this zone to protect this species. The third goal consisted of detect if there are some differences in pattern in terms of space use, and to see if that could be related to different behaviors or habitat preference groups. Then we aim to compare horizontal movements between reproductive and resting periods. And finally, we want to understand the reasons of some extraordinary vertical events and to see if they could be linked to some spawning deep events. This is the study area. So it is a small no-taxon of 3.9 square kilometers within a very large protected area that is called the Southwest Alentejo and Vicentine Coast Natural Park in Portugal. Inside this no-taxon, we have several habitats. We have the reef shore shallow rocky reef that are present all around the coast and spread towards the outside of the area, but also some rocky outcrops around the Marginal Islands, and the offshore area are more of a sandy bottom. 34 white seabream were captured inside the no-taxon using a line and a hook, and they were an internal acoustic tag was inserted, an acoustic tag from VEMCO, which is called V9P, and which comes with a depth sensor, which is quite. The area is surrounded by 32 omnidirectional receivers that are all around the zone, and that log the information into an internal memory. 7 of them couldn't be used because of some damage issues. And the study period started end of January 2000, late April 2020. So we have 1 year and 2 months of data. When we retrieved our data, 4 fish with very few detection were removed from the data sets because to avoid biasing the results. And all the process was done with RStudio. To understand if there were different behaviors driving to different patterns in space use, a classification system was adapted from the one used by my fellow student Carmela Abello, De Benito Abello, sorry. And we adapted this classification system and separated from the resident fish. And then within the resident fish, we identify 2 main clusters of detections that were leading to 2 habitat preference groups. One on the western part of the zone, which is the island preference group, and one on the eastern part of the zone, the nearshore preference group. In order to define the area usage, we first calculated the position averages, which is based on weighted means of the number of detection at each receiver with a 30-minute time interval. Then we calculated the minimum convex polygon, or what you call the MCP, which is basically the zone that encompasses all the position of the fish that are detected. And this is used as an indicator of the fish dispersion throughout the zone. After that, we decided to estimate the activity areas using the kernel utilization distribution, which is basically a probabilistic approach that estimates the area of probability of finding a fish. So 95% CUD is representing the home ranges, and the 50% CUD the core areas. In order to statistically compare the differences in space use between fish groups and period, reproductive and resting periods, generalized linear mixed models were used with the fish ID as a factor, as a random factor, and the Kruskal-Wallis test when we had few data. Then, when we plotted the vertical movements on the depth profile of each fish, we realized that they were showing some extraordinary vertical movements. So we decided to create some rules in order to allocate them. We selected all the data below the median depth of each fish tile in order to choose the depth threshold for each fish. Then we also use a delta depth threshold based on the 99% quantile of all depth variation together. And finally, based on that, we add the event duration threshold, which is the 90% quantile representing the 20 minutes threshold. And then we add the 24-hour rule to have a maximum duration threshold. A generalized additive mixed models was used with the fish ID as a random factor again, in order to study the count of events per day and to test some explanatory variables. So some that might be related to the reproduction, like the sea surface temperature, moon cycle, photoperiod, and months, but also two other that were related to the weather, the wave height and the wind speed. So now let's have a look at the results. When we look at the horizontal movements, here we see the two maps that represent the transient fish during the reproductive period on the left and the resting period on the right. And what is important to say here is that except from two fish, all the transient fish were present in the zone only during the reproductive period. So because of the small sample here, the small sample size, the statistical analysis was precluded. However, it's important to say that some fish that comes from other area come here only during the reproductive period. When we look at the resident fish, it's the same. We compare both reproductive and resting periods, and we see that for the resident fish, they spend all of their life cycle inside the zone. Their home range and core areas in green show that they have this basically the same during both periods. But what is interesting to note is the MCP that we can see here, which is higher during the reproductive period. It means that the fish move more and towards more offshore and towards deeper areas. Here we are looking at only the resident fish, but the two different habitat preference groups. We see that.
Sorry, I still saw the slide with transient.
Ah, really?
No,
and now?
Until now. Now I saw the.
Okay.
I don't know the residents. I imagine.
Exactly.
Sorry to interrupt, but you are commenting the residents, and I'm looking at the transient. Sorry.
Can you see now the island and nearshore?
Yeah. Now.
Okay. Sorry, there was a delay probably. So here we see that within the residents, both habitat preference group really show some site attachments that are really specific and distinct, that we can really distinct from each other. And what we see also is that the nearshore fish are really close to the border. So maybe it might mean that they are going towards other areas that are not monitored by the zone. But this is only an hypothesis that still needs to be tested. What is also important to say is that the MCP is significantly higher for the island preference group than for the nearshore. Here we are comparing for both groups the two periods, reproductive versus resting periods. And we see on the box plot here that the island preference group show a significantly higher MCP during the reproductive periods, while it is the contrary for the nearshore group. In other words, when we look at those maps, we see that the island fish are going more towards deeper areas, while for the nearshore fish, it is the contrary. During the reproductive period, they use a more restricted area. This is not really understood for now. Now let's have a look at the extraordinary vertical movements results. So here we see a time series graph that shows the number of events per day. And we see that the highest number of events are happening during winter and spring for both years. This is quite interesting because it really matches with the reproductive period in this area. But we may wonder, is it really related to spawning events? Actually, 71% of the GAM model deviance is explained by those variables I told you about that might influence the reproduction that are related to the weather. So now I'm going to show you the variable that shows the highest percent of significance. So here you see the sea surface temperature on the left and the photo period on the right, and both show an inverse relation with the number of events. When we have a closer look at the sea surface temperature, explaining 15.8% of the deviance, we see that the highest number of events are happening between 15 and 17 degrees, which interestingly match with what we found in the literature to be the range of temperature that triggers the reproduction. For the photo period, it is the events are more abundant when the photo period is low, corresponding to winter days, and when it's increasing, corresponding to spring days. For the months, it is deviant, and as we said, winter and spring months are the highest abundance. And for the wave height effect, it is also interesting because it's explained 11% of the deviance, and we see that the bigger the wave, the highest number of events are happening. And when we look at this graph, it really looks really related. However, we see that some days show some high peaks that are not actually, that does not seem to be related to that. So both phenomena, the reproduction and the big waves, are likely to happen during the same time of the year. So that's why it's difficult to distinguish their effects, to separate them from each other. But we think that they might be linked to a mix of both. So even though this study is not complete yet, we can still draw some conclusion. Regarding the horizontal movements, we really proved that there were very different behaviors within the same species and within a small area. Also, the fish showed some very high site attachment. During the reproduction, it was quite complex, but we realized that some fish are coming only during the reproduction periods within this area. For the island preference group, they use wider area during the reproductive season, while for the nearshore, they use a more restricted area. For the vertical movements, they are happening more often during winter and spring, which might be related to reproduction and environmental conditions. But still, further analysis still need to be done. And among them, we thought about studying the horizontal extraordinary events, but also investigating the vertical events and see if they occur in particular areas, to see if we could match both of them. I would like to thank all the people that made that possible, and especially Barbara, my supervisor, but also Ines, Luis, and Carmela, that were of great help, as well as two of my professors, Eric and Margarida. Thank you very much for your attention.